I'm going to be talking extensively about applications of a nuclease inactivated artificial form of CRISPR and what we can do with that in bacteria, yeast, human cells, etc. To do that, though, I need to talk a little bit about there we go. All right. I need to give you some background. So the title for my talk today is Repurposing CRISPR as an RNA-Guided DNA Binding Platform. We're going to talk briefly about an introduction. I'm going to tell you about, sort of similar to what Dana did, about previous work that had been done on programmable DNA binding proteins from sort of the late 80s on. I'll tell you why there was a need for something like a repurposed CRISPR system. And then I'll go into, in the second, tomorrow predominantly, the uses that we've used this repurposed CRISPR dCas9 system for as a means of turning gene expression on and off in prokaryotes and eukaryotes, as a means of directly editing the epigenome, for imaging the genome in living human cells, and then even newer applications for imaging or editing the transcriptome. And then lastly, for mutating the genome in a way that's independent of DNA double-strand breaks. So at UCSF, we were aware of Jennifer's work. We were sort of in awe about how easy the CRISPR system might be to use, but we had the audacity to wonder, like, well, what else could you do with this CRISPR system, and could you use this as more than a nuclease? And so I think as has been touched on, the beauty of CRISPR relative to previous methodologies is that it's really simple to use. All you need is one protein and one short chimeric guide RNA. And these two things, when co-expressed, search across the genome, bind to its target site with some variable specificity, as Dana just highlighted, and then make a double-stranded break. But we knew from Jennifer's very earliest work that this protein has a defined set of domains that are required for its double-strand break forming activity. And so these are the RUVC and HNH domains. And Jennifer had shown very, very early on in the first repurposed CRISPR paper that these two domains could be mutated and that this protein in vitro could still bind to DNA in the presence of a guide RNA. And so that suggested that the double-strand break-forming activity of a CRISPR system was separable from its RNA-guided DNA binding activity, and presented the possibility that we could use these systems as more than just a site-directed nuclease. And so that's what I'll talk about today and tomorrow. We think of this as sort of a broken set of molecular scissors. So this is an article from The Atlantic where somebody broke these nice functional scissors, and we've done more or less the same thing with Cas9. And so this data, this project, as Jennifer highlighted, was started sort of before I got to UCSF, and was originally the brainchild of a person named Stanley Shi, or Lei Shi. He's at Stanford now with his own lab. He was a grad student here at Berkeley with Adam Arkin, and was sort of jointly co-advised by Jennifer during his time at UC Berkeley. He then started a independent fellows position at UCSF right as the CRISPR craze was going on. And Stanley's a synthetic biologist. He had the foresight to ask, Jennifer had already shown that we could make mutations to Cas9 and that this protein would still bind to DNA. Stanley had the insight to wonder, well, what else could we use this for? And so he was the first to make these two inactivating mutations to Cas9 and then ask what we could do with this in a cell rather than just in a test tube. So what I hope to convince you of over the next sort of 2.5 hours is that we can use a nuclease deactivated version of Cas9 that we call dead Cas9 as an RNA-guided DNA binding platform, and that we can use this system to, in theory, deliver any protein domain to an arbitrary sequence of DNA in any cell, prokaryotic or eukaryotic. So that's the preview of what we're going to talk about. To get there, though, we need to talk a little bit about how we figured out how to do this in human cells. And so really what we initially focused our efforts on was whether we could use a repurposed CRISPR system to turn gene expression on or off. And I'll describe why we did that over the next slides. But rather than the very general question of can we deliver any protein domain to any arbitrary sequence, we asked a specific question of can we just turn transcription on or off, because we were leveraging a history of synthetic biology that had shown that this kind of approach could be used to control transcription. And so before we dive into that, I want to back way up. So why do we really care about CRISPR? So CRISPR is a means of studying of biology in general, right? But it's not that we particularly care about, well, this may be a little bit of heresy, but we don't particularly care about the DNA sequence that we're modifying. I mean, the genome is important, right? But the genome is just a record keeper, or essentially it's a set of instructions. What we really care about is the functional output of the genome. So what we're talking about usually when we care about perturbing cells, we care about perturbing mRNAs and proteins that the proteins have some function that can be revealed either by changes to the DNA or changes to the RNA or changes to the protein. And so in principle, we could make changes at any level and use that to study how the genome specifies cell function. So I'm fairly focused on human biology, so I like to use this picture of a baby as an example of what I'm talking about here. Every one of us in this room has started as a single-cell fertilized embryo and then has grown into a full-size human adult. All of us in the room have at least 200 cell types. The genome in every one of your cells, excluding mutations associated with replication, should be the same. And so it's not really the genome that specifies you as a unique person. It's the transcriptome and the level of protein expression in each cell that specifies cell function, that then contributes to organ function, that makes you who you are. And so in that way, what we're doing when we're studying biology is we don't really necessarily care about the genome. We just care about being able to perturb biological function. And so another way to think about that is what we really are doing when we ask most biological questions is we're asking how do cells know when and how much of each mRNA to make that's encoded by each gene in the genome. And so what we think about when we think about using dCas9 is essentially can we use dCas9 instead of Cas9 to perturb transcription instead of the genome and ask how genes function to contribute to biology. And another way of thinking about this is that the genome is like this piano. The piano is the same in any room across the world. There might be like grand pianos or baby grand pianos or whatever, but the keys and the notes are the same. And it's really what makes music is not the piano itself, but the way that the keys are played by a pianist, and not just which keys, but the intensity of the key strokes and the order in which they're played. And that's sort of the way you can think about how transcription orchestrates the output of the genome to specify function. This can be done at a variety of different levels in biology. So we can do, in cell culture, cell biology, where we ask how individual genes encoded by these HeLa cells are associated with specific cell behaviors and correlate that to normal physiology or to disease. We over time have become more sophisticated as biologists, where we've started to use more and more sophisticated organoid type models to model human biology, including in this paper they're looking at using ES cells or iPS cells to model the human brain. Or there's a rich history of using model organisms to study eukaryotic biology, and would highlight the mouse here as a model of mammalian physiology and disease. But in any case, what we really want to know is how do the genes encoded in the human genome or in the mouse genome specify function? So I've been talking a lot about trying to convince you that there's a genome and there's a transcriptome, and we can study biology by either studying how the transcriptome acts to influence function, or by how the genome acts to influence function. This all is related to the idea that there's a classic central dogma in biology that the DNA of any cell encodes genes which are transcribed into RNAs. These are then translated into proteins, and the genome stores the information inherited in the genome of a cell, the RNA is just a messenger, and then the proteins actually do the functional business. This isn't always true. We know that there are RNAs that have effector roles in cells as well, the ribosome being a classic example of that. But largely it's true. And so as a means of using dCas9 to study biology, what we really want to do is perturb transcription. We know that we can perturb transcription in two ways. You can generally inhibit transcription, or you can inhibit transcription in a specific way. So there's two different types of protein effectors that control the output of the genome. General transcription machinery and transcription factors. Transcription is very complicated, so that it's not a simple process to turn the information encoded in the genome into the correct set of RNA molecules that specify cell behavior. We know that the RNA polymerase, whether is fairly conserved across all life, so it seems to be a molecular machine that evolved once and then has been sort of repurposed as life has gotten more, I don't know, more complicated is the right word, but has changed. And so that's seen here in these crystal structures. You have these universally conserved portions of RNA polymerase. There are additional adapter subunits that have been added as we've gone from bacteria to archaea to eukaryotes. But generally, the basal transcription factor machinery that produces RNAs from the genome is conserved across all life. And so that'll be important later when we think about how we're using dCas9 to actually control transcription. More interesting to me is not the basal transcription factor machinery, but rather sequence-specific transcription factors. So again, I'm just really fascinated by this idea of asking how a cell knows how much of each RNA to make and when to make it. And this is a complicated problem that people have studied for a really long time. It is a problem that is sort of variable in scale across life. So I like to use this chart as an example of very simple eukaryotes versus more complex eukaryotes, and just the range of eukaryotic biology that that exists naturally in regulating genome expression. So this is a mycoplasma. It's a sorry, I said eukaryotic, I meant life. This is a very, very simple prokaryote. It's got one of the smallest genomes known. It's a single cell organism. The genome size is about 500,000 bases. It encodes 517 genes. And this form of life has no sequence-specific transcription factors that we would canonically annotate. So every gene is just sort of expressed in a more or less, not in an unregulated way, but there are no specific DNA binding proteins that specify gene expression in the way that we canonically think of them. If you move towards more complicated forms of life, like the eukaryotic Cerevisiae, or this is really representative of sort of intermediate, generally level of complexity. There are bacteria that are this complicated as well, where we've seen an expansion in genome size to about 12.5 million base pairs. You see that the number of genes has increased about tenfold, but the number of transcription factors now has gone up. Well, you can't calculate a fold change here because you can't divide by 0, but by a lot. So yeast now have evolved a set of sequence-specific transcription factors that encode complicated regulatory programs that are required for different lifestyles in this yeast, whether it's sporulation and meiosis or adaptation to different carbon sources or what have you. These transcription factors are absolutely critical for yeast surviving in the wild or even in the lab under a variety of conditions. So if this is sort of just all hardwired together, now we have a more complicated set of almost software instructions where you can plug and play different software programs using these transcription factors as master controllers of genome output. We're more complicated, but this is representative of any mammal species, basically. We have at least 200 cell types that make up humans. Our genome size is, again, much bigger than a simple eukaryote. We have about 3 billion base pairs. We have only about a 4-fold expansion in the number of protein coding genes, but we have about a 10-fold expansion or more in the number of transcription factors encoded by our genome. So the number of genes has increased, but what is increasingly appreciated is that it's not necessarily the number of genes, but the regulation of those genes that specifies organism complexity. So we have a huge expansion in the number of transcription factors that are required for specifying different cell types and coordinating genome output across our different cell types that make up organs and ourselves. So how does this all work? And I promise I am getting to a point here. The transcription, I hope I've convinced you that transcription is complicated. There's an interplay between general transcription factors, including RNA polymerase, and then sequence-specific transcriptional activators, repressors, mediators, and coactivators. There's feedback loops, so things can get dynamically rewired on and off. There's positive and negative feedback loops. There are multiple regulatory sequences encoded by the genome, both proximal and distant. There's also, in eukaryotes, local chromatin state that influences gene expression. So there are very old observations that the eukaryotic genome is covered in chromatin, and this chromatin has different densities, and this influences gene expression. To zoom in even further, though, we know that promoter structures in eukaryotes are complicated. So the genome is not a 2D linear sequence of information, right? It's a 3D sequence of information where there are loops and there are higher order domains that control genome output. And so to control transcription in a natural setting, each gene has promoters and enhancers that contain binding sites for specific transcription factors and general transcription factors. And these are just some canonical accepted definitions for what promoters and enhancers are and contain. And I'm going through this only because we're going to talk over the next, predominantly tomorrow, about how we can use dCas9 to edit the epigenome and to control transcription. And to think about why that's useful, we need to have some common language within this room about what enhancers are and what they do to control gene expression. So I'm going to talk tomorrow quite a bit about epigenome editing at either enhancers or promoters. And so just if you can, this is really a reference slide. If you get confused tomorrow, the slides will be online or are already online. You can refer back to that. But it's not just DNA sequence. There's also proteins that are associated with the DNA sequence with the genome that have very complex chromatin modification code that controls both the structure of the genome and gene output. Okay, so that's all. That's the introduction. That's basically, if I have one point, it's that the genome is complicated. But the regulation of the genome is perhaps even more complicated than just the primary sequence. There is a complex interplay in prokaryotes and eukaryotes between proteins, RNAs, and DNA that governs the output of the genome. So probably since people have realized that DNA is, composes the inherited portion of life, people have been interested in figuring out how that is decoded. And so what I'll talk to you today initially about is our early efforts to understand how proteins decode the information encoded in the genome, and then how that was used to or leveraged as a synthetic biology tool for building tools with which we could control transcription. And these the lessons that I'm going to talk about over the next couple of slides are really, they are fundamental to everything that we'll be talking about in the course. So Dana has talked you through a series of tools where people are using either constitutive or regulated promoters to express Cas9. All of those promoters came from this kind of work which I'm going to talk about in the next couple of slides, where people analyzed endogenous or artificial promoters and endogenous or artificial transcription factors and learned how they worked, and then we can repurpose those as tools for biotechnology. So this may seem like sort of old, but you're all still using these tools right now, whether you know it or not. And so I'll talk initially about artificial transcription factors, and then we'll dive into Cas9. So again, the genome encodes lots of specific sequences that govern the output of the genome. Those are bound by transcription factors. Transcription factors are modular proteins. They all have their characteristic aspect to all transcription factors is, or most transcription factors, is that they have a DNA binding domain. So they're modular proteins. They have a DNA binding domain. They can either specify depending on the context in which they bind, the transcription should be activated or inactivated, and that is largely contextual and depends on the local state of the chromatin and their co-regulatory binding partners. But the point of this whole slide is basically that they are modular. So there's always this DNA binding domain. There's sometimes a dimerization module. Sometimes there's an activation module or repression module. The details vary depending on the protein. But the key point here is that people realized that you can usually split the DNA binding activity from the effector activity. And so this allows us to use synthetic biology to piece things together arbitrarily and encode different regulatory functions. So I want to go back all the way to the early 80s, which is at that point we knew that DNA was bound by proteins and that these proteins in some way specified gene expression. But there were two competing models at the time. One was that these proteins just bind to DNA and change the structure of the DNA in a way that allows for RNA polymerase to engage with a gene and induce gene expression. This would be sort of a biophysical model that has turned out to be more or less incorrect, although there are specific cases where that can be true. And then the model B was that that the genome encodes specific binding sequences that recruit proteins that then specify some function, not by changing the structure of the DNA, but rather by bringing in other proteins that encode additional activities. And so there was a series of experiments done in the early 80s in E. coli using lambda phage that essentially derived as, there was an operon, these phage can integrate into the E. coli genome, and then they can either be lysogenic, meaning they just are present in the bacteria and they replicate with the bacteria as an integrated phage, or they can be lytic. And the switch between lysogenic and lytic phases of phage biology was controlled by just a single protein. So what people asked was, can we decouple the ability of this master controller of lysogenic versus lytic biology to see whether DNA binding encodes the activity of this protein or whether there's some other function that we can separate from DNA binding. And so they made a series of mutants and put them in. They derived this one mutant where you could still get binding, but that the gene expression of this reporter that was integrated in the phage was no longer expressed. So they knew that this was the first experiment essentially that said that transcription factors are modular proteins that encode multiple behaviors, one of which is DNA binding, but that can be separated from the ability to control transcription. And so this was an elegant idea, but phage are really simple, and E. coli is also quite simple, and people were not convinced that this would be true in eukaryotes. So in yeast, around the same time, people were trying to ask the same question using the glucose-galactose nutrient switching system or sensing system, and they knew that this is a nice system for studying transcription because it's fairly simple. In the presence of glucose, you have Gal1 transcription is off, and you have this Gal4 protein that is bound to the Gal1 promoter, but it's inactivated by the presence of this Gal80 gene. When you add galactose, galactose binds to Gal3 and induces release of Gal80. The Gal4 protein, which is bound to the promoter of Gal1, then induces transcription of this Gal1 gene. And so essentially, the system was simple enough that people thought that they could ask whether transcription was controlled by DNA binding or whether it was controlled by the presence by transcription factor ability to recruit additional proteins. And so they went about trying to figure this out in a way that is sort of the same as we still do for a lot of cell biology. They decided to make a series of truncating mutants and internal deletion mutants, similar to what Jennifer showed for defining the H&H anti-CRISPR activity interaction. They used a reporter, which was this LAC-Z gene, which can be easily scored, and then asked whether they could separate in a eukaryote the transcription factor binding activity from the ability to induce gene expression. And so the wild type gene shown here both binds to this promoter element and is able to activate beta-galactosidase activity. If you make a series of truncation mutants, they were able to show that the DNA binding property of this protein is confined to just the extreme end terminus, and that they could delete the vast majority of the protein and still retain the ability to activate transcription, and eventually settled on the minimal required portions of this protein for DNA binding and activation of transcription being confined to the N and C terminus. And this was really the first sort of synthetic biology experiment that showed that we can construct artificial non-natural transcription factors that contain both a DNA binding activity and an ability to activate transcription. Really, a lot of this experimental framework has just been reused again and again and again to repurpose more and more DNA binding proteins and different domains for controlling transcription in either prokaryotes or eukaryotes. I like to show this is from a review a couple years later, but I think sometimes we lose how far we've come in science because the actual data is this. It's essentially just a hand-drawn cartoon of what they did with arrows to score, so no real numbers, but it's all correct, and it's been extremely influential in both the fields of synthetic biology and understanding how transcription works. The field moved quickly beyond that, so there was a desire to ask whether we could use completely artificial. So these are natural proteins that are found in yeast, but what we'd like to be able to do, there was already a realization in the 80s that if you could use artificial proteins that you could where you could specify the DNA binding motif, you could use this to control cell behavior. And so the same groups very quickly started to use other bacterial and eukaryotic proteins and fuse these together, making artificial chimeric proteins, and then see whether you could turn transcription on or off of endogenous genes or reporters. So this was the first, to my knowledge, artificial transcription factor where they fused the DNA binding portion from this LEX-A protein to the transcription activation portion of this Gal4 protein. And then they were able to show that in a way that was not specified by normal nutrient sensing, they could turn transcription on of this gene in the presence of glucose, indicating that they had rewired the normal activity of Gal4 to turn on transcription, but using the LEX-A DNA binding domain. This works in human cells as well. It's remarkably very, very similar. So most human transcription factors have a DNA binding domain and a transcription activation or repression domain. And so around the same, or a few years later, people started to fuse these same yeast transcription factor DNA binding domains to domains that they suspected could activate or repress transcription in human cells. So this paper had an example of a Gal4 ER fusion or a Gal4 glucocorticoid receptor fusion. And you can see if in the presence of estrogen mimetic or glucocorticoids, you can induce the activity of these artificial transcription factors turning on gene expression. It's a little bit hard to see here, but basically you're going from gene expression being off to gene expression being on in the presence of estrogen. So this was a really useful early example of the kind of thing that you could do, where you can construct these completely non-natural transcription factors, and if you know enough about the biology, you can make them chemically inducible. So we're turning on gene expression in the presence of an artificial transcription factor only when we add an exogenous ligand. So this is a direct sort of academic grandfather of the split Cas9 systems that Dana talked about, where people have used various domains that bind small molecules and are induced in the presence of a small molecule to induce Cas9 activity. The same thing was done here with a Gal4 VP16 fusion. VP16 is a protein domain from a herpes virus. And I show this only because we're still using VP16. We haven't found anything better than VP16 for activating transcription. So this was the first paper that showed that you could fuse the yeast DNA binding domain of Gal4 to the herpes virus VP16 domain and then activate transcription of this reporter artificially. And this was also at the time used to figure out how the spacing of transcriptional activation worked relative to gene transcription start sites. So this is what I mean when I say that many of you are probably using these systems whether or not you realize it. So if I would guess that for anybody in this room who's worked on mammalian systems, you've probably used or thought about using a TET-on or a TET-off system. These systems are artificial fusion protein of the TET repressor DNA binding domain to the herpes virus VP16 domain. So depending on the orientation you're talking about, in the presence or absence of doxycycline, you can turn on or off transcription of these artificial promoters. So you have multiple binding sites for the TET-R protein upstream of a minimal CMV promoter, usually, which drives your reporter gene of interest. And so these have been around for a long time, but they're still incredibly useful. We can also turn transcription off if we just fuse other domains on. So we're gonna talk about turning transcription off quite a bit. And the way we turn transcription off is usually by using these scaffold domains called CRAB domains that recruit other enzymes. And so as early as the early 90s, we had already started to assemble effector domains that can control gene expression in human cells, either using the VP16 domains to turn transcription on or the CRAB domains to turn transcription off. And this will be echoed again and again in a variety of applications. So here's a more current application where we're using our artificial transcription factors to turn gene expression on or off. Here we're using, this lab was interested in using artificial transcription factors to define factors that are required for heterochromatin formation, which is important for controlling gene expression during development. And they knocked into the OCT4 locus of a mouse binding sites for either this FKBP fusion transcription factor or sorry, this ZFHD1 binding site or a GAL4 binding site, and then they have these fairly complicated transcription factors which are artificial fusions that dimerize in the presence of either rapamycin or this plant hormone called abscisic acid. So you can independently control recruitment of either a domain that induces heterochromatin formation or a domain that activates transcription to ask how the formation of heterochromatin is coordinated by various silencing and activation activities. And you can ask fairly complicated questions about co-occupancy and order of addition and timing during ES cell models of differentiation and heterochromatin formation. But essentially, I just want to use this slide as a takeaway of saying that these fixed sequence artificial transcription factors are still being used, and they're still incredibly useful. This is a second example of that, where you can actually go beyond this with synthetic biology and use GFP as a scaffold for specifying different activities with the same, where you can use GFP as a scaffold to specify multiple activities in different cell types depending on where GFP is expressed. So this lab was interested in leveraging the many GFP reporter mouse strains that are available publicly, and then activating transcription dependent on the expression of GFP within a cell. And essentially, the details here are not important, but they are using fixed sequence artificial transcription factors and a bridged GFP reaction to turn on or off transcription in different neural cell types, and using that to dissect different brain properties. But the problem with all of the examples that I've shown you so far is that these proteins are only able to bind a single DNA sequence. So if you want to turn on or off expression of a gene, you either need an artificial promoter or you need to knock the DNA binding sequence into the genome. And so that really limits the flexibility of the applications that you can use with these TET-R or GAL4 systems. The field moved forward sort of in a parallel way with the genome editing field, where some of the There were very early papers after the recognition that we could use these human zinc finger transcription factors as an artificial way of controlling transcription. And Dana showed you data from around the same point where people fused zinc finger proteins to the FOC1 nuclease. At about the same time, they fused these same zinc finger proteins to CRAB domains and VP16 or 64 domains to turn gene expression on or off of endogenous genes. So these are unmodified genes encoded by the human genome, and you can construct an artificial transcription factor that turns gene expression on or off in a parallel way to mutating the genome, actually. The issue, as Dana alluded to, is that the engineering for these zinc finger proteins is quite complicated, and there's only probably a handful of labs in the world that are capable of doing this correctly. Dana being one of them, and Sangamo, and a variety of other people. But similarly, as soon as the tail code was cracked, people started to use tail proteins to control transcription. So like Dana said, these tail proteins are transcription activator-like effector domains. They have this 34 amino acid protein sequence that recognizes 2 base pairs. If you use fairly complicated synthetic biology to string together a bunch of these 34 amino acid repeats into a repetitive protein, you can assemble a protein that can recognize a 20 base pair sequence and append a CRAB domain or a VP16 domain to that, creating an artificial transcription factor that recognizes your sequence of interest. And this was used to turn on or off transcription. This is actually, I find this sort of interesting. This is Feng Zhang from the Broad's early work. So he was hired at the Broad to do TALEN work, and he predominantly was using tails to control transcription before he got into CRISPR. So this is, he's been thinking about these things for a long time. This is work from Keith Jung's lab, or also from George Church's lab. If these names sound familiar, it's because people hopped from one genome editing or one artificial transcription factor technology to CRISPR very quickly. So as late as 2013, Keith Jung's lab was using tail fusions to activate gene expression in the human genome. So these are examples where they're using multiple tails in combination to activate these different endogenous genes encoded by the human genome. The problem with all of the things that I've talked about over the last however many minutes is that as for genome editing, all of these methods rely on a protein-DNA interaction, and you have to re-engineer the protein every time you want to turn on or off a new gene. And so that was really, really slow and expensive. I have a table in a different set of slides that shows all of the genes in the first sort of 8 or 9 years that had been mutated, activated, or repressed using zinc fingers, and it's not a huge list. You can fit it on one slide. So these were not trivial experiments to do. They remain robust technologies if you care a lot about a specific target. Zinc fingers and tails are a great and very specific way to either mutate the genome or turn on or off transcription. But for research purposes in the average lab, the field was stuck here where there was obvious utility, but there was the cost to manipulating gene expression with these technologies was too high. And so people predominantly were using RNAi or cDNAs to manipulate gene expression. So that's sort of where the field was. There was this desire to be able to control the output of the genome, but we didn't have an easy way to do it. CRISPR came in, and most people are focused on mutating the genome to control gene output. But what I'll show you is that we can also control transcription directly. So I showed you this slide before, but essentially, Stanley wanted to know whether we could use dCas9 to control transcription in bacteria. And so he cloned a bacterial version of dCas9 that could be expressed in an E. coli cell. And just asked a very simple question of, if we express dCas9, what does it do? Does it bind the genome in a living human cell? Does it interact with RNA polymerase? Does it control transcription? Can we use this as an RNA-guided DNA binding platform? And to begin to ask some of these questions, He constructed this dCas9 sgRNA system. You can transfer this to E. coli, and he asked a very simple question of what happens if we target a fluorescent RFP reporter with 6 different guide RNAs targeted to this reporter, and just measured the expression of RFP if you targeted dCas9 to the gene body of this RFP reporter. And so I think what he saw, it still is sort of amazing to me that this works as well as it does. What he saw was that if you target dCas9 to this reporter in a strand-specific way, you see a very, very strong effect on transcription. So these results are for these sgRNAs targeting the non-template strand of this RFP reporter. What he saw was that if you target dCas9 close to the transcription start site of the non-template strand of this reporter, you turn transcription of this reporter off by about 100 to a couple hundred-fold. And so this, I really don't think any of us expected this result. This is a very strange result, to be honest, in that both bacterial and eukaryotic chromosomes are coded in proteins. Most of those proteins don't block an elongating RNA polymerase. But in this setting, we have a non-natural protein being targeted to the genome, and it in some way very robustly blocks an elongating RNA polymerase. And so we still honestly don't understand all of the ways that Cas9 and dCas9 interact with the genome and with other proteins that are occupying the genome, including, in this case, RNA polymerase. But what was immediately obvious was that this was going to be a useful tool. So we were able to very robustly control transcription in bacteria, and this could have applications in all kinds of, either research in pathogens or in biofuels for metabolic rewiring and things like that. One of the things that Stanley noticed was that the ability to control transcription is contextual. So if you target dCas9 far from a transcription start site, it has less of an effect on transcription than if you target it close to a transcription start site. And then targeting the non-template strand has a robust effect on transcription where targeting the template strand doesn't. So there's something about the biophysics of the Cas9, dCas9 interaction with RNA polymerase that influences the ability, its effect on transcription. But this kind of activity is only useful if your ability to control transcription is also specific. And so Stanley, together with Matt Larson and Jonathan's lab, did an RNA-seq experiment where they looked at the transcriptome of E. coli where they were expressing an sgRNA or no sgRNA targeting RFP. This is a log 10 plot, so it's a little messy, but you can see that when you express a guide targeting RFP, RFP goes down, and most other transcripts encoded by the cell don't change by a whole lot. And so this, together with a lot of subsequent work, has shown that our ability to control transcription in bacteria is very highly specific. Mechanistically, because the result was so surprising that dCas9 blocks transcription very efficiently, we wanted to know whether this was whether we could measure this in another way. And so fortunately at the time we had a method called NetSeq where we could tag RNA polymerase and then pull down to map RNA polymerase relative to the genome, so you can measure spatially where transcription is happening across the entire genome. So if we do this and we express dCas9 plus or minus an sgRNA targeting this same RFP reporter, we see that in the absence of a guide, you see RNA polymerase is coding the entire gene body of this RFP reporter. So these are these peaks here, where these are normalized counts that are representative of RNA polymerase transcription. And then if you express dCas9 plus a guide, you see that dCas9 binds right here, and RNA polymerase initiates transcription and extends about 150 base pairs, and then is completely blocked right at the edge of the dCas9 binding site. So this is pretty good evidence that RNA polymerase cannot, in E. coli, remove dCas9 bound to the genome, and that's the basis for our ability to control transcription. How am I doing on time? How much time do we have? 2 minutes?
You're dominantly going to 12:30. You could go a little longer.
I'll be right on time. Okay, so what else can we use this tool for in bacteria? One thing we showed very early on is that we can use dCas9 not just to block RNA polymerase, but we can actually use it to block specific binding sequences within promoters and control transcription that way. So if you target dCas9 to the promoter of the same RFP reporter, there were known sequences within the promoter that are required for transcription, and we see that if you block this -35 element, which is really critical for transcription initiation, you robustly block transcription. In this example could be in part, it's impossible to decouple whether this is due to RNA polymerase binding or initiation, but either way we showed that we could use dCas9 to compete with an endogenous gene binding site at a promoter. More usefully for synthetic biology applications, we were able to show that you can use chemically inducible systems to generate inducible control of transcription, where if you add an inducer, so this is, sorry, let me back up. We constructed a system where you have an inducible promoter that drives expression of dCas9. If you don't have the inducer present, transcription is proceeding normally. You add the inducer, you see that we turn gene expression off. You wash the inducer out, you see gene expression comes back on. So this indicates that we can dynamically control gene expression, and also that dCas9 isn't doing something permanent to the genome. It really is an inactive form of Cas9. It's not mutating the gene at a low level, and that these kind of examples have been used in a lot of papers subsequently for metabolic engineering in bacteria and biofuels. We can multiplex these systems in both prokaryotes and eukaryotes, so we can get additive effects on transcription in E. coli if you target 2 guides to a gene. This is the fold change in repression. You can see that both have a strong effect on their own, and that increases if you have 2 guides. Or you can target 2 different genes. So this is an experiment where you can target either RFP or GFP or both. And you can see the pictures correspond here, where here you're targeting RFP, here you're targeting GFP, here you've turned both off. And so multiplexing is especially useful for metabolic engineering. And so that will be, has been used for a variety of applications for producing different metabolites that are desirable for different, either lines of research or bioproduction. This system in bacteria has been extended now to a couple additional bacteria. So this is a recent paper in Bacillus subtilis where they used more or less the exact same dCas9 system to study antibiotic resistance. They showed that similarly you can generate inducible systems where you can control transcription dynamically. So here's expression of the gene in the absence of an inducer, and then if you add xylose, gene expression goes off. Yeah, off. This is a cleaner RNA-seq experiment where they're measuring transcription in 2 populations of cells that do or don't express a guide targeting RFP, and you can see that if you target RFP expression goes off and no other genes change. This is the same NetSeq assay where you can show that dCas9 is indeed blocking and elongating RNA polymerase, and that's the source of our ability to silence transcription. And then you can similarly show that you can control expression of multiple genes. And I show this just as a secondary validation of what was shown in E. coli, but also because we know a lot about E. coli, and our ability to extend these systems into additional bacteria are really where the utility of this toolbox will become most manifest. And I know there's a lot of work published and unpublished right now extending dCas9 into tuberculosis. Tuberculosis has been a really hard bacteria to work on, and there are really exciting things in terms of medical applications that are going to come out of our ability to control transcription in prokaryotes. And then, so I'll end with this slide. This is one of the final figures in this paper. If you're curious to read more and you care deeply about Bacillus subtilis, but essentially they were able to use dCas9 to identify the direct molecular target of an antibiotic that they were interested in. So the principle underlying this is that if you repress the direct molecular target of a compound, that has an outsized effect on the cell's sensitivity to the compound. So essentially you decrease the intracellular concentration of a protein, and cells are more sensitive to a compound if it's the direct molecular target. So they showed that with a genetic screen here, and then they did a bunch of follow-up to show that this was true. But I hope that this has applications in antibiotic research in tuberculosis and Pseudomonas and other medically relevant pathogens. So I'll end there. That's this was the necessary but boring introduction. And then I really like the bacterial stuff. I think that these kind of tools are going to be incredibly useful over the next couple of years. And tomorrow we'll pick up with the rest will be all human. So if you care more about human stuff, that's what we'll talk about tomorrow morning. And thank you.
Questions for Luke?
It's only working in bacteria. Why is it not? If it's right, it's only working in bacteria. Why is it not transferable to eukaryotic cells?
So it does work in eukaryotes. So in eukaryotes, dCas9 does block transcription by interfering with RNA polymerase elongation. It only decreases transcription by like 2 to 4-fold rather than a couple hundred-fold. I think that's largely because, as I showed in one of the early slides, RNA polymerase is quite different between prokaryotes and eukaryotes. We have many additional accessory subunits that people think predominantly act to render RNA polymerase more processive. Our genes are, instead of 1 or 2 kb long, or they're like, including introns, hundreds or megabases long. So we need RNA polymerase to, with very high fidelity, be able to transcribe across hundreds of thousands or millions of base pairs of DNA. And if it gets blocked after it's already transcribed, 100,000 base pairs of RNA, that's really energetically costly to the cell. I don't have a perfect answer for you other than it is what it is, and for some reason eukaryotic RNA polymerase is more processive.